Good morning everyone and welcome to my YouTube channel. So here today we are going to be looking at macromolecules. So what are macromolecules? So macromolecules, the word macro simply means large. So these are large molecules that are made up of atoms. So the first class of macromolecules that we are going to be looking at is carbohydrates. So what are some of the things that you're supposed to know under carbohydrates? The first thing that you're supposed to know under carbohydrates is that the general formula for carbohydrates is it has CN, H2N, and ON. That means to say the number of carbons is equal to the number of oxygens. The number of, so CH2N, ON. So the number of hydrogen is double the number of oxygens. You will understand this more as we proceed. Okay, so just to shed more light, let's say if you are looking at glucose N is equal to 6. So glucose will be a C, where there is N we have 6, H2 where there is N we have 6, then we have O 6 like that, which will give us C6, H12 or 6 like that. So this is the formula of glucose like that. So now the carbohydrates have got a different range of functions, such as, so firstly, carbohydrates are used in respiration. So number one, the first function is that carbohydrates are used in respiration. So I can just put respiration. So under respiration, there is the release of energy from sugars such as glucose. So you might be familiar with reactions such as C6H12O6 plus. If it is burnt in oxygen, it produces carbon dioxide and water. Now it is not only carbon dioxide and water plus our adenosine triphosphate, which is the energy that is used to power many cells. So carbohydrates are used in respiration. The carbohydrate here, this one is what we are going to call our glucose like that. So this is an example of a carbohydrate, which is a monosaccharide. So the other function of carbohydrates is that they are used in storage of energy. So under storage of energy, many carbohydrates can be stored in forms such as starch. So carbohydrates can be stored in plants. So starch in plants acts more like as a reservoir, or rather a storage form of glucose. So this can be used as a storage form of energy. In animals, we have something like glycogen. So when there is a lot of sugar in the blood, it is converted to glycogen. So the sugar we might be talking about, most commonly glucose, fructose, or galactose, it is converted to glycogen. This glycogen is the storage form of glucose in animals. So this is very, very important, as it acts as a reservoir. In case the animal does not have food, it can easily convert that food back again into glucose. Then the other storage form of glucose that we might have is alright, for now we can just look at those. So carbohydrates have got many ranges of functions. So this was number 2. Then number 3, the function that we are going to talk about is it is useful in the structure, formation of different structures. So these structures, we might have something like, number 1, we might talk about cellulose. So cellulose is the form of carbohydrates that is used in making cell walls. So cellulose is used in making cell walls. So cell walls are made of this thing that we call cellulose. The other form of the structural form of carbohydrates that we might talk about is what we can call chitin. So chitin is just the cell wall. It is something that helps make the cell wall of fungi. So we are going to dive deeper into their structures and what are some of the things that are unique for them. So you will also find glucose in part of the nucleic acids, which helps in passing down genetic information. So now let's move on to what are some of the things that we expect under this coverage. So let's look at our monosaccharides. So monosaccharides, these are the small, simplest sugars. So the word mono means one. So these are the simplest sugars. So the examples that we might have under monosaccharides that we can have things like glucose, we could have things like fructose, we could have things like galactose, like that. So these are monosaccharides, among other things. So monosaccharides, remember the carbohydrates have got CH₂O. So beginning with our number of carbons to be 2, we are going to be calling them diose. So the name will come from the number of carbons that are there. So if we have 2 carbons, di means 2, so we are going to have diose there. The example that we might have here is a glycoaldehyde. So we might have glycoaldehyde. We might have glycoaldehyde like that. So like this. So this is an example of a diose. So the structural formula might look like something like this. So remember, it is CN, which is the 2 of them, H2N, which will be 2 times 2, it will be 4. Then we have O like that, or 2 like that. Then what we are going to do is that to draw the simple structure of this, it's going to look like this. So just begin with your carbon bonded with a double bond, oxygen like that, draw an hydrogen like that. Then since from there you're just going to begin. So you have 2 carbons, you're going to write another carbon. Then on your right, you will just write your hydroxide like that, then your hydrogen. So you can just spell water for the first one, water. Then last but not the least, we are going to just have hydrogen. So as you can see, we'll have 1, 2, 3, 4 hydrogens, and number of oxygens 2. So this gives us C2. Then we have our H, 1, 2, 3, 4. Then we have our O2 like that. So this is the simple structure of a diose, which is a carbohydrate or a monosaccharides containing 2 carbons. The other structure that we might look at is what we call a triose. Tri means 3, which means to say we are going to have a 3 carbon sugar like that. So this is going to look like something like this, double bond O like that. So remember, we should have 3 carbons, so 1, 2, 3 all together. Then we are going to begin. We have our OH like that. Remember, we first have our OH, then we are going to have our H, then we are going to have our H like that, then we are going to have our OH. It's more like they are interchanging. OH comes this side, then we are going to have our hydrogen for the last part like that. So this one is our C3, so H should be 6, then we have our O should be 3. So let's count. We have 1, 2, 3, which is 1, 2, 3, then hydrogens 1, 2, 3, 4, 5, 6. Then our oxygen, we have 3 of them. The example of a triose that we might have is a glyceraldehyde. So this is our example that we might have. Then we can have what we call a tetrose. So tetrose, so initially it's supposed to be a tetra, which means 4. Now if we combine it with an O, we remove the A, then we are going to have a tetrose. So a tetrose is a 4 carbon sugar. So same thing, to draw this structure, we are going to do something like this. Double bond O there, then we have our hydrogen, then we have 1, 2, and number 4, like that. Then we are going to do the same thing. We have our OH here, then we are going to have our hydrogen, interchange it. So hydrogen should come this side, our OH this side. We are going to finally have our OH this side. We have our H and our H, like that. So this is our C4H8O4. So you can count 1, 2, 3, 4, 5, 6, 7. Oh, we are here. 1, 2, 3, 4, 5, 6, 7, 8. So there should be 8 hydrogens, like that. So 8 hydrogens, 4 carbons, and 4 oxygens, like that. So now let's move on to things such as the pentose. Now here we are going to be using the ring structures. So under the ring structures, for some we are just going to be using ring structures. For others, we are going to further explain them using these types of structures. So ring structures occurs when these sugars are more like put in water, when they exist as molecules, therefore they form rings that might look like something like this. So now let's draw certain structures. So let's look at what we are going to call pentose. So pentose are 5 carbon sugars. So the 5 carbon sugars that we might have are what we are going to call a ribose, which is a part of RNA. We can also have what we call a deoxyribose. This one, the deoxyribose, is found in DNA. So now when we draw certain structures, we are going to have something like this. So drawing the ring structure of this, we are going to have our O, like that, then draw the pentagon. So that's the meaning. You draw the pentagon like that, then we are going to get something like this. Then on top, we are going to put your OH like that, then you have your H like that. Then on this end, always just put your CH₂OH like that. Then here, you are going to have, down here, you are going to have your hydrogen like that. Now here, you are going to have your hydrogen. Even here, you are going to have your hydrogen. This is for pentose sugar. Then the only difference, ribose, you should have this. So both of them here have got an OH. Then the only difference is here. If you have OH here, it simply means to say you are looking at a ribose, which is the sugar found in RNA. On the other hand, if you have this structure, so same thing that we were doing. Now the only difference between these is that, so here we shall always have the same thing, which is our OH. Now here you can have your H. So you can just think of it as your Dio means, just think of it as Dio means 2. So you should have the same thing for this side. So this is the only key difference between the two. So this is the deoxyribose, this is the ribose, like that. So you should know how to draw these structures, and also don't forget what I said. So ribose is a sugar found in RNA, then deoxyribose is the sugar that is found in DNA. So this key difference is very, very important for you to distinguish between the two. So in other words, for ribose, at the 2' of the carbon, there is an OH. So the primes are, if you have oxygen here, you begin to count like this is carbon number 1. So where there is a bend, that means to say there is a carbon. So carbon number 1, carbon number 2. So this is the 2 prime. So carbon number 2 is 2 prime, like that. So at the 2 prime, for ribose there is an OH. For deoxyribose there is just H here, which is down here. That is the key distinguishing figure. So we have, if this is 1 prime, 2 prime, we have 3 prime, we have 4 prime, then number 5 is here. So this is our 5 prime, like that. We can put 5 prime. So these are primes. Then for deoxyribose, at the 2 prime there is hydrogen, like that. So let's move on to our next structure, which we are going to call our hexose. So from the word hexagon, so hexose are 6 carbon sugars. Examples that we might have here for hexose is we might have glucose, we might have fructose, then we might also have galactose. So these are some of the hexoses that we might have. So now let's dive deeper. So before that, let's just look at what are some of the disaccharides formed. So disaccharides are formed when 2 or more of these combine together. So let's say if you have glucose plus glucose, so di means 2. So di for this one is di means 2. So we should have 2 monomers, which are glucose, fructose, and know that when they combine, glucose plus glucose will give you what we call maltose. Then if at all we have the galactose combines with glucose, then what is formed is just get the last letters from here. So what is formed is what we are going to call lactose, which is found in many things. So this is mostly prominent in substances such as milk. Then when we look at maltose, this is found in germinating seeds. So don't forget that lactose in milk, maltose in germinating seeds. Now on the other hand, if let's say our glucose was to react with our fructose, then this one is produces what we are going to call sucrose. So we are going to have our sucrose, or should I spell SU? So we have our sucrose, like that. So these ones are found in substances such as sugarcane. So they can be found in substances such as sugarcane. So in another video, we can compare and contrast the fructose, galactose, and glucose in terms of structure. But for now, let's now focus on what are some of the compounds that are made by glucose. So glucose might be, firstly, glucose might be alpha glucose. So this is for alpha glucose, or it might be beta glucose. So there are 2 forms of glucose. We might have alpha glucose, then this is our beta glucose, like that. So let's dive deeper into their structures. So when we are looking at our alpha, what I like to think about is the position of the hydrogen. So alpha, just think of the hydrogen. Alpha, the hydrogen is on top. So this is how we draw them. So we are going to have the shape of an hexagon. Since it's an hexose, we should have something like this, like that. Then we are going to combine each and everything. So let me just rub here. So we have something like this. Now what you're going to do is that, just as we did for those others, we are going to have our CH₂OH here. Then here we are going to have our H. So if you extend this, you are going to have your H. Think of it as an attraction. If here there is H, here there should be our OH. So if you know that, then here we are going to have our hydrogen like that. Then here, since we have our OH there, we should have our H here. Then down here we should have our OH like that. So then here we are going to have, here we always have, for both of them, we will always have, yeah, just missing a bit. Yeah, okay, so here we are going to have our hydrogen like that. Here we are going to have our hydroxide like that. So this is for both our alpha and our beta like that. So now the only difference is this side. So alpha, I told you to say, think of just the hydrogen atom. The hydrogen should be on top. Alpha means on top. So now the only difference is, for alpha glucose, there will be an hydrogen on top. Then here we are going to have OH like that. So this is the hydrogen will be on top like that. So this is how the arrangement is. Beta, on the other hand, everything is the same, but the only difference is here, in which on top we shall have our hydroxide. Then down here we shall have our hydrogen like that. So this is how the structure looks for our alpha and our beta glucose like that. Now when these combine with each other, they are able to produce structures such as amylose and amylopectin. So let's look at what are some of the structures that are formed when the alpha glucose combines. So when the alpha glucose combines, we are going to have, if two alpha glucose combine in the 1-4 glycosidic bond, glycosidic meaning to say they are just the bond between the carbon atoms, or rather the bonds that is formed when these sugars combine together. So when these combine in this 1-4, so this structure is a linear structure. So let me explain how it goes. So when these structures come together, so our main interest for this reaction is just this side. So when these structures come together, this is what is formed. So we are going to have, so the reaction that these undergo is what we call a dehydration reaction, in which water is released. So how do we release water? We know that water is equal to our H₂O. Our H₂O is water, so meaning to say we need 1 part of hydrogen and 2 part of hydroxide. So we can take this like that. So if we take this, what we have is that this is taken away, it becomes into water. Then if we combine them, we are going to have, so for the sake of simplicity, we are going to just show what is bonding there. So we could have something like this. Yeah, so this is what we have. Hydrogen is remaining. Then here we are going to do this and put our oxygen there, combine it with like that. Then we have our hydrogen also on top. This is taken away. Then we have the structure. It's looking like this. So we have an oxygen there. So this is what we have like that. So this structure has just been drawn for the sake of simplicity like that. So here we have each and everything, but just to explain, this is what I am drawing. So this is a 1-4 glycosidic bond. Remember, we calculate primers. We said this one, the first carbon from oxygen going clockwise. So 1, number 2, number 3, number 4. So this is carbon number 4, then this is number 1. So this is where the bond is taking place. So this is a 1-4 glycosidic bond. So this is what we have. So a 1-4 glycosidic 1-4 glycosidic bond. So this is our main thing of interest. So now this 1-4 glycosidic bond, so as you can see, this is a straight chain. So if we have another one, it will be an O, then we are going to have a structure that will look like this. So it will continue like that. So this is what we call a 1-4 glycosidic bond, like that. Now when we look at this, this is what we call amylopectin. Amylopectin is formed by the structure in which the glucose, alpha glucose, combines in the 1-4 linear chain. So as you can see, this is a straight line, it will not bend like that. Then when we look at or did I say amylopectin? No, this is amylose. So this is amylose. Then when we look at amylopectin, so amylopectin is now the one that is formed. Amylopectin is formed when glucose, which is still the alpha glucose, combines in a 1-4 and a 1-6 glycosidic bond. So in this type of a structure, there are what we are going to call branches. So the amylopectin will branch. So we are going to have this type of a structure. Remember, we are just dealing with alpha for now, then we are going to go to our beta. So this is the structure that we are going to have. So for the sake of simplicity, if we have one carbon like that, then it bonds like that. So this is a 1-4 glycosidic bond like that. Then we are going to have something like this. Here, what we are going to get is, so remember, this is a 1, this is a 1-4, so we have oxygen there. Then what we are going to get is that, so this is the structure. So remember, here we had a CH₂ON like that, OH, sorry. Now we are going to have a branch. Another carbon might join here. Now what is the structure that is formed? So just in case it came in a test, you have to just finish the structure. I've taught you how to draw the structure, but for the sake of being fast, I want to more like finish fast. So we have another carbon might join here. So as you can see, this is prime number 1, number 2, number 3, number 4, number 1, 2, 3, 4, 5. Then here we should have carbon number 6 at the 6 prime here. So what happens is that we are going to have a structure in which our hydrogen from here will combine with this to produce water. So when there is the production of water here, it means to say we have removed this structure. So this type of a bond will look like this. So the 1-6 is there. Then here, if we remove this, we are going to have a structure that is going to look like this. So we have something like this. So remember, we removed our hydrogen, so we are remaining with oxygen here, then our CH2. Then here it is going to bond with that oxygen. Let me move it a bit. So we have this type of a structure. So as you can see, this is carbon number 1, this is carbon number 6. So this is a 1-6 alpha carbon. So alpha, remember here it's HOH like that. Then what we are going to get is that we have a 1-4 and a 1-6. So this is the structure of amylopectin. Now amylopectin is easy to digest as because of these branches, meaning to say enzymes can easily work. They have greater surface area, meaning to say they can work at many points at the same time. On the other hand, for amylose, it is not so easy to work on, since these are just linear chains, meaning to say enzymes do not have a lot of points to work on. So we might have structures that might be formed as a result of this So these structures, the amylopectin, will now further branch on to produce what we call glycogen, which is the storage form of glucose in animals. So this is formed by a combination of 1 to 4 glycosidic bonds and 1 to 6 glycosidic bonds. So now the general structure of amylopectin, I spelled glucogen, no, glycogen. So amylopectin and amylose form an helix structure, which is this structure that looks like DNA, like that. Then let's look at a bit of our beta glucose. So when the beta glucose combine, so we have beta glucose. When they combine, it might look like something like this. So remember, under beta glucose, the one that is on top is our OH. Then we have H there. Then we have all those sorts of things. So for this one, let me just redraw the structure so that you can remember. So this is our O, like that. Then here we had our H, we had our OH. Here we had our we had our H was on top, so we had our C. We began here CH₂OH. Then down here we are going to have our H there. Then H will correspond with our OH. Down here we are going to have our H, like that. Here if we have our H, down here we are going to have our OH. Just to remind you, then here we are always going to have this H on top, OH. So this is our beta glucose, in which the hydrogen is down here. So when this combines with another one, this is the structure that is formed. So if we have something like this, so if we have something like this, then the structure that is formed, we are going to end up with, so already here it is this side. So here we have our hydrogen, here we have our OH. Be careful. So it is this side where it's changing, where we have on top it's OH, but this same structure drawn again will look like this. So that's the first. Then you are going to find out to say, remember how the structure is formed. So your hydrogen is, your oxygen is the one that can form a double bond. So therefore, you need to expose an oxygen. So there's no way for you to do this and forget. No, it is not possible, because if you remain with your OH has got a valence of 1, meaning to say it can only bond with one thing. So you need to expose one of the oxygens. So what you're going to do is that you are going to grab one of the hydrogens, combine it with an OH, like that. So this releases your water, like that. If water is released, what we are going to have is this type of a structure. So we have the simplified structure, which will look like something like this. Then remember, we are remaining with our oxygen there. We are remaining with our oxygen. So here we have our hydrogen, like that. Now to put the oxygen, we can put the oxygen here, but to put the oxygen at the middle, we are going to have a bend like that. To put our O like that, then we have something like this. So this is what we have for our beta, like that. So this is how our betas combine. So this is the only difference. It's more like it's forming a Z, or rather an N shape. So structures that are formed by beta glucose doing this is what we can have as our cellulose. So our cellulose is the combination of the beta glucose, in which they are bonded in a 1 to 4 glycosidic, so glycosidic linkages. So these types of bonds, where they bonds together in that straight chain, now a lot of them. So this is what we might have. So here down here hydrogen and all that. So we might have this structure repeating itself throughout like that. So this is what we call the cellulose, which is a 1-to-4 glycosidic linkage like that. Then the other structure that might be formed is what we call is what we call chitin. So chitin is formed in which we have, so remember, cellulose, this is a component that is used in animal cell walls, plant cell walls. So they arrange themselves in this order. Even down here, let me show it like this. So these structures will now be able to combine together. They will be able to merge. So these are held together by what we call hydrogen bonds. So now, when we look at chitin on the other hand, this is responsible for the formation of the cell walls that are found in what we call fungi. We might also have what we call crustaceans. Crustaceans, we are referring to animals such as the crabs. We might have the crabs. Should I say sea? So we might have the crabs like that. We might have lobsters. So those are crustaceans. So the structure is the same as this. Now the only difference is that at the 2' there is a protein. At the 2' of carbon, meaning to say if we have there one number 2, carbon number 2, there is a protein, which is this is our protein. So yeah, so this is the protein. CH3 like that. So this is the protein that is found. So even here we should have this same protein. So it is formed by the beta, so you can know by the Z like that. Yeah, thank you very much for watching. Hope you would like, subscribe and share for more. See you in the next tutorial.